For this video, I will be presenting and give brief explanation for my DNA reflection. So, there are two strands of DNA which run in opposite or anti-parallel directions to each other. These strands are attached to each other throughout their lengths via the bases on each nucleotide. There are four different bases associated with DNA: cytosine, guanine, adenine, and thymine. In normal DNA strands, cytosine binds to guanine and adenine binds to thymine. When bound together, the two strands form a double helix structure. DNA synthesis is initiated at particular points with the DNA strand known as origins, which have specific coding regions. These origins are targeted by initiator proteins, which go on to recruit more proteins that help aid the replication process, forming a replication complex around the DNA origin. Multiple origin sites exist with the DNA structure. When replication of DNA begins, these sites are referred to as replication forks. Within the replication complex is the DNA helicase. This enzyme unwinds the double helix and exposes each of the two strands so that they can be used as a template for replication. It does this by hydrolysing the ATP used to form the bonds between the nucleobases, thereby breaking the bond holding the two strands together. DNA primase is another enzyme that is important in DNA replication. It synthesizes a small RNA primer which acts as a kickstarter for DNA polymerase. This enzyme is ultimately responsible for the creation and expansion of new strands of DNA. So, DNA is made up of millions of nucleotides. These are molecules composed of a deoxyribose sugar with a phosphate and base or nucleobase attached to it. These nucleotides are attached to each other in strands via phosphodiester bonds to form a sugar phosphate backbone. The bond formed is between the third carbon atom on the deoxyribose sugar of one nucleotide known as the 3 and the fifth carbon atom of another sugar on the next nucleotide known as the 5. So that's all.